Welcome to Profound Knowledge.
Hello, this is about the antigen presenting cell and how it interacts with the T cell. This is the heading, antigen presenting cell and its interaction with the T cell. So this is all about the adaptive immunity. In the adaptive immunity, initially we have the antigen presenting cells, or initially even before the antigen presenting cell, you have a simple macrophage or any immune cell like B cell, dendritic cell. They will actually interact with the antigen that has come and into the body, that has actually entered into the body. And what happens is, once after engulfing the antigen and then after processing the antigen, the cells, macrophages or any cell that has actually got exposed to that particular antigen will turn into an antigen presenting cell. Here this is the macrophage which has turned into an antigen presenting cell or an antigen presenting macrophage that is now interacting with the T cells. So this interaction basically occurs and stimulates the T cell related immunity or the cell mediated immunity. So this is about the interaction between the antigen presenting macrophage and the T cell. So in T cell, lot of process will be occurring. Actually what happens is, after the antigen presenting cell is activating the T cell will be releasing the interleukin 2, or many cytokines are released. In that, interleukin 2 is the main one which we are going to discuss. So after the release of the interleukin 2 from this T cell or the helper T cell here, what is happening is, interleukin 2 released by the helper T cell will be actually acting on the same T cell and it will further lead to some other processes. So for our convenience, what I have done is 2 different T cells. So this is also one more T cell. So this is a macrophage, this is a helper T cell, this is also same type of the helper T cell. This diagram is basically indicating about the antigen presenting cell and how it is interacting with the T cell and what are various steps in the T cell activation and the release of the interleukin 2, and further how the cell mediated immunity will be taking place and what are the different drugs. This is about the classification of the immunosuppressant drugs and how these drugs are going to inhibit various steps involved in this antigen presenting cell interaction with the T cell and the cell mediated immunity, how they are going to inhibit this cell mediated immunity. So let us go through the process. The antigen presenting cell will be actually interacting with the T helper cell. How is, first we have gone through that the macrophage will be turning into a antigen presenting cell. That is, it will present the antigens over its surface with the help of major histocompatibility complex 2. MHC2 is major histocompatibility complex 2. So with the help of MHC2, over the surface of the antigen presenting cell, you have the antigen, that is the red color one, that is presented. And now this antigen presenting cell and the antigen over the surface will go and interact with the helper T cell. This black cell is nothing but the helper T cell. The helper T cell will be possessing a T cell receptors, that is TC or the green color one is nothing but the T cell receptor. So now the interaction between the T cell receptor and the MHC2 with the antigen will be there. So after both the cells interact, what happens is, the cell signal should be sent inside into the T helper cell. That will be helped by the Cd3 cofactor or the Cd3 co-receptor, that is actually a protein present in close vicinity or present very closely to the T cell receptor. And there is all that is the blue color one which will send the impulses or which will send the communication towards inside of the T cell receptor. Then there is also one more component just beside the T cell receptor. You can see this black color one, that is nothing but the CD4 component. This CD4 component is also a co-receptor that is present just beside the T cell receptor, which will strengthen the interaction of the T cell receptor with that of the macrophage. Now what is happening once there is interaction with the macrophage? then what happens is there will be activation of the T cell receptor. So T cell receptor gets activated, that will further interact with the inside of the T cell with the help of this CD3. Then what happens is there will be activation of the phospholipase C and tyrosine kinase. See here, phospholipase C and tyrosine kinase system that will be activated, and after the activation, what happens is there will be stimulation of calcineurin. See, let us see what is actually happening with the phospholipase C and tyrosine kinase. Here I have elaborated. We all know that the secondary mechanisms for the G protein coupled receptors, in that we have gone through, like this tyrosine kinases are present. Kinases basically help for phosphorylation of any enzymes. So there will be activation of the tyrosine kinase here. The TCR is getting activated, that will activate the tyrosine kinase, which is present intracellularly in the T cell. And then you have the phospholipase C, which will be phosphorylated by the influence of this tyrosine kinase. And this phosphorylated phospholipase C will further act on the phosphatidyl inositol bisphosphate, and that will be converted into inositol triphosphate and diacylglycerol. Inositol triphosphate will be acting on the calcium channels, and it will actually rise the levels of the intracellular calcium inside the T helper cell, whereas diacylglycerol will be acting on the phosphokinase C, and it will activate the system inside. Next thing is, what this actually calcium level, increased calcium level is doing plays a major role. So the raised calcium level will be going, the calcium goes and interacts with the calmodulin. CAM is calmodulin. So the calcium calmodulin complex will be formed. Now this calcium calmodulin goes and actually interacts with the calcineurin. Calcineurin is a phosphatase. It is an enzyme, phosphatase type of enzyme. So what it does is it will cleave the phosphates that are present over compounds. So it further goes and it will interact with the N-fat, which is already having a phosphate. So the phosphorylated N-fat will turn into non-phosphorylated form. So N-fat here is nothing but nuclear factor of activated T cells. So now this activated T cell will be having this N-fat. So N-fat, which is phosphorylated, will be turning into N-fat without a phosphate under the influence of this calcineurin. So this N-fat, which is not having the phosphate group, will be able to cross the nuclear membrane and will be able to enter into the nucleus. So there will be translocation of the non-phosphorylated N-fat into the nucleus. It goes into the nucleus and then further interacts with the DNA, and it will be activating a gene, that is NF-kappa B. So the NF-kappa B gene is the one which is actually responsible for the transcription of the mRNAs and formation of the proteins that are nothing but the cytokines. So there will be release of excess formation of cytokine will be there. The formed cytokines will be released out from the T cell. So what is that we understood now finally? The antigen presenting cell actually interacts with the T cells on the T cell receptors, and it further stimulates the release of interleukin 2. So now interleukin 2 has been released. The released interleukin 2 will show autocrine activity, that is nothing but it will be taken up by the receptors, interleukin 2 receptors that are present over the same cell from which it has been released. So it goes and acts on the receptors that are present over the helper T cells. Actually that will be acting on the same T cell or any other T cell that is present closely to the T cell that has actually released this interleukin 2. Now let us assume that it is acting on a other T cell just beside it. So what is happening is this interleukin 2 goes and acts on the interleukin 2 receptors that are present on the same type of T helper cell. This is the T helper, and then now what is happening is there will be activation of the interleukin 2 receptor, and which further goes and stimulates a mTOR, which is a kinase, mTOR, the full form is nothing but mammalian target of the rapamycin. So it has got its name from the drug rapamycin or the sirolimus. So now this mTOR is the major internal factor inside the cell that is responsible for the further activation of the genes, and then there will be proliferation of the T cells. So more number of T cells are formed. The T cells formed will further interact with the antigen presenting normal cells. That is, what happens is the infection that has actually entered into the body or the antigen will be also acting on the normal cells that are present in the body, and the normal cells also process the antigen and they display the antigen over their surface. So that kind of antigen presenting cells will be acted upon by the T cells that are actually proliferated. These are nothing but the T killer cells. So T killer cells go and act on the antigen presenting cells, and then this antigen presenting cells are lysed off or they will be killed, leading to the death of the antigen or the destruction of the antigen. So this is the process what has been actually occurring. Now let us go through various drugs that are actually helpful as immunosuppressants and how they are interacting in this process. So first classification of drug is calcineurin inhibitors. In the calcineurin inhibitors, you have drugs like cyclosporin and tacrolimus. Cyclosporin, you can see that the triangle is the cyclosporin, will be interacting with a protein that is called as cyclophilin. Philin indicates like loving, so that is it is cyclic compound loving protein. Now what is happening is cyclosporin goes and readily interacts with the cyclophilin. You can see that the triangle there is the cyclophilin. So both of them complex. This cyclosporin cyclophilin complex goes and inhibits the calcineurin. So the calcineurin will be inhibited. It causes inhibition of calcineurin. Actually, it will inhibit the calcium calmodulin complex and it will be further inhibiting the calcineurin, and further interleukin 2 secretion from the T cells will be inhibited. So that is the mechanism of action of cyclosporin. Next drug in the calcineurin inhibitors is tacrolimus. The other name for tacrolimus is FK506. So because of this name FK506, the actual protein on which this tacrolimus is acting is nothing but FKBP. FK is nothing but the tacrolimus itself, the name of the tacrolimus. BP is the binding protein. So FKBP is nothing but FK binding protein. So tacrolimus acts on the FK binding protein and it will further inhibit the calcineurin and further inhibiting the release of the interleukin 2 from the T cells. So from the helper T cells, interleukin 2 release will be inhibited by both calcineurin inhibitors, cyclosporin as well as the tacrolimus. Next class of drugs are mTOR inhibitors. So mTOR inhibitor is nothing but the sirolimus and everolimus. The sirolimus is also called as rapamycin. Here one thing you should remember is the tacrolimus, which is a calcineurin inhibitor, whereas sirolimus and the other drug is everolimus are the mTOR inhibitors. Both of them are also sounding something like tacrolimus, but their mechanism of action is different. Though their mechanism of action is different, they are going to actually act on the same protein. So tacrolimus, sirolimus and one more mTOR inhibitor, everolimus, all of them, they actually bind to the same FK binding protein. So all of them, they bind to FK binding protein, but their mechanism of actions are different. Tacrolimus, by binding to FK binding protein, inhibits the calcineurin, whereas sirolimus and everolimus, by binding to FK binding protein, they go and inhibit the mTOR. So here also you have its separate name. mTOR is nothing but the mammalian target of the rapamycin. It is a kinase which further activates some enzymes that are responsible for the T cell, and that will go further and interact with the nucleus and with the DNA, and then they are responsible for the T cell proliferation. So here T cell proliferation will be inhibited by the mTOR inhibitors. The next class of drugs are antiproliferative drugs. Antiproliferative drugs here are majorly the anti-cancer drugs, which you can remember like methazacyclo. Methazacyclo. Here my is nothing but the mycophenolate mofetil, clo is chlorambucil, meth is methotrexate, az is azathioprine, and cyclo is cyclophosphamide. So you have myclo, meth, aza, cyclo, all are basically anti-cancer drugs. What they are actually doing is, we know like in the cancer there will be excessive unwanted proliferation of the drugs which can be inhibited by the anti-cancer drugs. They are serving the same process here. They inhibit the T cell proliferation. So you can see like mycophenolate and azathioprine, they are nothing but the anti-proliferative drugs mentioned here. They go and inhibit the T cell proliferation. And glucocorticoids. So glucocorticoids or the corticosteroid drugs, prednisolone is the example here. So you can see like corticosteroids, they actually interact. Corticosteroids are highly lipophilic compounds. They are able to cross the cell membrane as well as the nuclear membrane. They are able to go and interact with the nuclear receptors present inside the nucleus. So here they go and interact with the NF-kappa B and they inhibit the cytokine production and the interleukin 2 production from the helper T cells. As well as they also have one more mechanism of action. They actually interact with the macrophages and they inhibit the expression of the major histocompatibility complex 2 over the surface of the macrophages. In such a way, they actually inhibit the interaction between the macrophages and the T cells. And the next class of drugs are biological agents. So we have biological agents, in that we have the TNF alpha inhibitors. In the TNF alpha inhibitors, like their target is nothing but they go and act on the macrophages. The macrophages, after getting activated, they secrete the TNF alpha, and this TNF alpha that is secreted from the macrophages will further go and act on the TNF alpha receptor 1 and receptor 2 that are present over the surface of the neutrophils, fibroblasts and some endothelial cells. Also, some of these receptors are present in the soluble forms in the serum are also there. So what is happening is, after acting on the TNF alpha receptor 1 and receptor 2, the TNF alpha will be amplifying the immune inflammation. That will be done by releasing the cytokines and some enzymes like collagenases and metalloproteinases. So all such activity will be inhibited. So TNF alpha that is actually released by the activated macrophage will be inhibited by the TNF alpha inhibitors. Next one is interleukin 1 receptor antagonists. Activated macrophage also releases the interleukin 1, and this interleukin 1 will activate the T helper cells, and that will be inducing the production of the interleukin 2. Now, so the T cell immunity will be following. So interleukin receptor 1 antagonists like anakinra will inhibit the interleukin 1 receptors over the surface of the T helper cells. Next thing is interleukin 2 receptor antagonists. Interleukin 2 receptor antagonists are nothing but this basiliximab and eculizumab. Here MAB indicates monoclonal antibodies, M for monoclonal, AB for antibodies. So monoclonal antibodies, they are basically monoclonal antibodies. They actually inhibit the interleukin 2 receptor over the surface of the T helper cells. They inhibit the expression of the genes, and then excess of T cell proliferation will be inhibited. Next thing is anti-CD3 antibodies, like anti-CD3 drugs like muromonab CD3. So muromonab CD3 go and inhibits the CD3 co-receptor that will be actually responsible for the communication after the interaction of the macrophage with the T cell receptor. The CD3 is responsible for the communication with the interior of the cell. And so the activity that is carried out inside the cell will be all inhibited, and the release of the interleukin 2 will be inhibited. So the muromonab CD3 will be inhibiting, it's an antibody. Monab indicates indicates monoclonal, AB indicates the antibody. So these are monoclonal antibodies that are acting against the CD3 component. They go and bind to the CD3 and they inhibit the activity of the CD3. And next thing is polyclonal antibodies. These polyclonal antibodies, they are actually going and inhibiting the expression of the excess of T cell proliferation, and T cell activity will be inhibited by them.